So think about what questions you want to ask. We're going to do this sort of Oprah style. So just give me a little signal if you have a question you'd like to ask, and wait for me to come with the microphone. One more thing, I've been asked a number of times, will these slides be available to you after? And this is for everybody, for our virtual audience as well. We will be rebroadcasting this entire webcast on our Facebook page for the next few weeks. Unedited raw footage, and you can go through it anytime. You can take screenshots and stuff like that of slides if you want. And in about a little over a month, I will be posting annotated videos for this. So we'll break it up. We'll have an immunotherapy page and a personalized medicine page. And if you look at our website, if you go and look at past roundtable programs, we have them for the last 3 years. We have them all recorded with notes so that you can go through it very quickly. You can read along with the notes and listen, or go to certain parts of it if you want. We also organize every question that's asked. So with that, if our speakers could come up, please. And like I said, Dr. Kumar, he's just going to join us at some point. These doctors, as you get to know them, they're always in meetings, they're always in calls. It's really remarkable that we even get them here at all. So who would like to start out with our first question? Don't be shy. Everybody's shy. Okay.
Hi, I hope I'm remembering this properly. Dr. Schroeder, you were talking about the CAR T cells, 90% experience side effects, 40 to 50% success rate. Did you have the same stats for the bispecific engagers?
Yeah, so the cytokine release syndrome can happen in bispecific T cell engagers. In general, it's a similar range in terms of the cytokine release, but what's reported is maybe a little bit less than the CAR T cells. It depends on the one thing about CAR T cells is that it's given, you can't take it back. The bispecific antibodies are a treatment that you can get depending on their half-life. The one case I showed, it's a half-life that's given every 3 weeks, so it hangs around in your system some time. And so if you get cytokine release with that type of treatment, it's going to continue. If you have a drip that's continuously dripping in, you shut off the drip, that side effect goes away. Can we predict who's going to get these major side effects? The cytokine release syndrome, I can tell you it's common. If I was looking at a patient in the clinic, I wouldn't be able to pick out the 20% that aren't going to get it and the 80% that could. Or 90%. So it's hard to predict. Sometimes the burden of the cancer itself does predict for the severity of that reaction, an inflammatory reaction. You're having the T cells revved up, they're attacking the cancer, or that antibody's binding the cancer and bringing in all the T cells to attack the cancer. In terms of response rates, depends on the study, on the treatment. Both bispecifics and the CAR T cell treatment have been reported to have patients who, like I showed, complete responses, no detection of 1 in a million myeloma cells. Those responses, at least in CAR T cells we saw in the earlier talks, can wane over time. And so the case I presented of 2 years out is not typical for somebody after CAR T that has relapse refractory myeloma. They might progress sooner, but that's why there's research to try and improve treatments to try and move these therapies earlier on in the treatment.
Hi, Dr. Borrell. My question's on the vaccines or the GVAX. Is there a specific time during the treatment that this is given? Like, is it during MCOS stage, during the maintenance, right after transplant, can it be given later? And also, how close are you guys to approving this with FDA? Well, how close, what stage of the trials are we? Are we like initial trials, or is this something that's going to be more advanced?
Okay, so in terms of so I think in general, the way vaccines are going to work, at least the way I think they're going to work, is if there's a very small amount of disease, or if the immune system of the patient is still relatively responsive. So the trial that we designed has been to do this for patients that are on a Revlimid-containing regimen, because we know that Revlimid can augment immune responses, that are in a complete remission. In that situation, we know that those patients aren't cured. Their disease burden is low, and they're on this immune stimulant drug called Revlimid. So that's the current trial. However, if that trial were to prove to be positive, the best case scenario would be to do it in any situation in which you either have a low disease burden or the patient hasn't really progressed. So theoretically, following autologous transplants, following CAR T cells, I think, potentially even following, bispecific antibody therapy if patients can get into remission. And I think the whole idea of smoldering and possibly MGUS would be as well very interesting. I think the problem from a clinical trial perspective is that those endpoints end up being very, very long. And so although that is the ultimate goal, because if we could get into an immune system before it becomes very beat up with chemotherapy or beat up by the myeloma itself, that is where ultimately I think we're going to have the biggest impact. We are still in the very early phases of this. And so when assuming a positive trial, we're talking about, in the best case scenario, 5 to 10 years before this could possibly be an accepted therapy that's FDA approved, assuming that everything goes well.
Question about treatment planning and sequencing. Because the immune system is so important to all these newer therapies, is there an argument to not do a chemo and stem cell procedure first, but instead to continue to treat with triplet or quad and save that and perhaps use some of the other immune based therapies and save the stem cell and chemo for a later time?
No, I think it's an excellent question and an open one in the field. Most of us actually do think that for patients who are transplant candidates, stem cell transplant actually does offer the most effective way of reducing the tumor burden and almost hitting a reset button on the immune system, if you will. Because as we spoke about earlier, cancers did not originate overnight. It took a long time before myeloma actually developed. And so what a transplant does is almost provide the reset button in that regard and achieves the maximal reduction of tumor burden. Having said that, there are studies ongoing to try to ask the question of whether or not things like CAR Ts could be tried instead of transplant or before transplant and things like that. Those are all clinical studies that are currently ongoing or being designed. The same is true for bispecific antibodies as well. So I think the ongoing studies will actually inform some of these questions in terms of what the timing is. But I at least personally do not recommend to my patients to not undergo a transplant because of the option of the future of CAR T therapy or bispecific.
If I could just add, so there was a big important French study that was published a few years ago that actually looked at VRD, which was at the time state-of-the-art therapy, sort of until disease progression versus a few cycles of that followed by transplant. And I think at the time that was an interesting study because the question began to surface: if we can achieve such deep remissions without transplant, do we need transplant? And I think the conclusions of that study was that despite the use of this highly effective therapy, there was still a deeper complete remission rate, a deeper MRD negativity rate. So that means even 1 in a million cells, there wasn't any evidence of disease, and a longer progression-free survival, so time to relapse. So as of today, standard of care for most patients should still be transplant. These other questions should be answered in the context of a clinical trial.
And if I may, one of the hard parts of what I do in meeting your needs is you have to balance with what is the situation today, what's the optimal situation today, with what's coming in the future. And today we're talking more about what's coming in the future. This is also, I think, a symbol or a sign of how myeloma is progressing so rapidly, because we can, trying to think of the word, it's not responsible anymore for patients like you to not talk about what's coming 3, 4, 5 years from now, because you have to start thinking about that, even if that's not available today. And that's a very different situation than patient meetings we used to have 20 years ago. So we have one question here.
Yes, for Dr. Vidge. When they talk about Bispecific or CAR-T, is there a criteria that says this patient should go one way or the other? And I was just kind of curious also, how many people in the trial program here now?
Yeah, so thank you for that question. So I think that's a question that is subjective to each patient. As answering your second question first, we have at least 4 trials with CAR T cells. We have about 4 trials with bispecifics available. And what is the best fit for a patient not only depends on the patient's situation, but also sometimes the criteria that the trial will allow patients with certain prior treatments, certain health conditions. So I think that when these all get commercialized, then it is going to be a learning curve as to how to sequence these treatments. With the CAR T cell, for example, we already have for the last 6 months a CAR T available. Now the problem is that there are serious production problems with that CAR T, and only there are probably 60 centers right now that are licensed to give the commercial CAR T. Each one of them can give probably 1 CAR T a month, perhaps. And then this situation will not resolve probably till next year. So I think these kind of things are going to be practical issues also that will determine which way you go. With bispecifics, we are seeing early deep responses. What we lack data right now is the durability of these responses. I think at our annual meeting, which occurs usually in December, we'll probably start seeing the first data on how long these responses last. It is possible that some of these may come close to CAR T cell responses, in which case then it becomes a question of what is better for a given patient. Practicalities with the bispecific, as you heard, you can give the bispecific literally within 2 or 3 days of the decision. With the CAR T, what happens is that for a commercial CAR T, insurance companies want you to negotiate a rate which they already know what they're going to give you for each patient. And so it takes them usually 2, sometimes 3 weeks to tell you, okay, we've signed a single patient agreement with Washington University. Now you can go ahead and give the CAR T. So I see a patient today, it will take me 2 to 3 weeks for my institution to, their lawyers to sign off on the CAR T. Then it will take about a few days to a week to schedule you for apheresis. And the manufacturing time with delays can sometimes, after the collection, can extend to 5 to 6 weeks. So you're talking sometimes with the CAR T, from the time I see a patient, it'll be 2.5 months before I can give it. And so some patients, that's not practical. So I think there's so many considerations.
In 2017, I was diagnosed with multiple myeloma, received a remission in 2018, and then last year in 2020, developed AML. In the treatment, is either dominant right now in the treatment, or am I just odd?
So I think that unfortunately secondary leukemias and secondary cancers are a well-recognized feature of our treatments. Now, even in the era before Revlimid, the drug you get for your transplant, Melphin, has about a 5% chance of producing pre-leukemia or leukemia 5 to 7 years out from the treatment. With Revlimid, we sometimes see not only acute myeloid leukemia, but also acute lymphoblastic leukemia emerge. And often that emerges a little quicker. And this is thought to be possibly a side effect of the maintenance with Revlimid. And we've done some, like other institutions, work on this, and there's a particular genetic abnormality, this p53 deletion, which characterizes a lot of these leukemias. So For the vast majority of patients, the transplant and Revlimid paradigm, I think, benefits them, but unfortunate few who develop these secondary cancers, obviously, you are, unfortunately facing a situation you thought you should avoid. So, but for the vast majority, it benefits.
Dr. Jadopkar, it looks like you're going to.
The thing I also wanted to add was that it's not a guarantee. We don't know for sure exactly what caused the leukemia in this situation. In many cases, there are some suggestions that there may actually be alterations in stem cells that give rise to both malignancies potentially. So I think this is still an unknown at the present time.
Maybe just to add to one more thing to that is, again, patients with myeloma, always there's an increased risk of second cancers. And that we've been seeing it over years, and now that patients are living longer and longer with the current treatments, it's probably we are seeing a little bit more than what we have seen in the past. So there's always that increased susceptibility, and maybe some of the treatments that we do, like transplant or the maintenance with Revlimid after transplant, may add on to that risk.
I would like to seize the opportunity to thank you guys, to thank you, all of you, okay, for what you are doing, and for the hard work, okay, that you've been putting to make people like myself, to give a chance, okay, to people like myself. My question is, in addition to conventional medicine, would you agree, okay, that the success of the therapy is paramount to the mental status of the patients?
Repeat your question.
Okay, okay, in addition to conventional medicine, would you agree that the success of the therapy is also dependent or is paramount, okay, to the mental status of the patient?
Balancing mental status and treatment? Okay, so how do you balance those? Mental health and treatment, that's a big one.
So I think the short answer is probably yes. I can tell you that they've done studies in breast cancer where they've looked to see this. People with a positive attitude, people that exercise more, people that are not depressed tend to do better than patients that have those things. Now, interestingly, also what has been shown in a lot of other cancers has been that if you're somebody that is not necessarily has an underlying depressive disorder, being given the diagnosis of cancer does not necessarily increase the percentage of people that are depressed. So, in other words, cancer doesn't make people depressed. If you're depressed, you'll be more depressed if you have cancer. And in general, people that are not depressed and have a healthier attitude tend to do better. So I think the short answer is yes.
Hi. Yeah, I want to second him. Thank you for everything you guys are doing. Very appreciated. I'm a Karma 2 trial patient participant, cohort 2, just about 2 years past Cardi, best I've been since diagnosis, so thank you for all your work. That leaves me with 2 questions, and you kind of talked about it this morning. Why do our counts linger so low for so long? And then also, what do we know about the durabilities of someone that's participated early in a CAR-T?
So I think that, again, the counts being low at the initial phase of the first several weeks are because of the fludarabine and cytoxin you get. The CAR-T and the cytokines they release probably also contributes. But some people have more prolonged cytopenias, and it's not totally clear whether it's that pre-existing marrow damage or something unique to the CAR-T process that drives that. And that is an area of active research. As to your question about how long responses last for people who get CAR-T early in their journey, I think that is something that is still being studied, and we haven't seen data emerge from these trials. People like you who are participating in these trials will hopefully give us this data in the next year or two.
maybe just to add to that, indirectly, I think we have some sense that the earlier might be better, and that's again based out of some of the longer term follow-up from trials in China, where those patients did not get exposed to the same array of medications that people in the US would have been exposed to, and there they seem to have found maybe it lasts for 2.5, maybe even up to 3 years, suggesting that people who have not become very, very resistant to current therapies, it might work even better.
There are other things that are coming out as well. So I think there's a lot of evidence to suggest now that it's sort of like an army. The more soldiers you have, the higher the likelihood it is that you win the battle. And so higher doses are doing better than lower doses. That's been shown. The kind of intracellular signaling of the CAR-T looks like it's predictive. So for example, right now there are 2 of them. One is CD28, the other one is 4-1BB. 4-1BB, when they look at these, what we call meta-analysis, all these data together, the 4-1BB tend to have better outcomes than the CD28 intracellular mechanisms. And the other one that's also evolving is that the extracellular part of this can either be made with a mouse background, a human background, or what's called a cameloid background, which interestingly, what they're using now are llama antibodies. And those llama antibodies, which is what the Janssen, or the Legend, I think BioLegend is Janssen, right? The Janssen CAR-T has this cameloid antibody, probably is also predictive of better outcomes. So there are a lot of other things in addition to, your disease status going into this.
One thing I would also like to say is what's often unsaid about CAR-T commercialization, which I think patients should know because they're going to be the advocates to change this. So for example, when you have a CAR-T commercialized, it costs today like $416,000, $420,000 a sticker price for the CAR-T infusion for in that range. Medicare has a reimbursement of $269,000 fixed for the CAR-T. So the thing is that Medicare will pay the institution literally half of what the CAR-T costs the institution. Then they tell the institution that you can make up for that by doing what's called this carve out for extra charges. They have terms for this, and the way you do that is you tell us what your cost of a CAR-T is, and you put it on your website, and you say, so for example, institution X will say the cost that it costs us to actually give the patient the CAR-T and everything is $700,000, and then they will be paid a proportion of that to make up for the lost cost. And so a lot of institutions are hesitant to put a very big markup on their website, and that's the only way they can actually be compensated for providing the services. There are institutions that for the same CAR-T have on their website a cost to patients of $700,000, and another institution charges $2 million for the same CAR-T. Now the institution that charges $2 million finds it economical to give the CAR-T because Medicare will pay them a proportion of $2 million. Whereas for an institution that is somewhat cognizant of its public image and charges only $700,000 will barely make even the cost of the CAR-T. And this is a really sad situation that Medicare has put this whole field in, that you can't even, without financial skullduggery, try to get CAR-T to patients. And this will possibly be a barrier in the future as these become more mainstream. Institutions may decide this is something that we are not financially able to provide, and we're not going to give certain CAR-Ts at all, and let the patients go elsewhere for their CAR-T because we don't even get reimbursed for what we are. This is unfortunately a sad side of medicine that we have to be upfront about, that these things are happening.
We got a message from our online viewers, and for everybody, speakers and everybody, please put the mic uncomfortably close to your mouth when you speak. Thanks.
I'd like to also thank all of you. This has been just fascinating. I am 6 months post stem cell transplant, so I was really interested about the healthy lifestyle, the Mediterranean diet, how do we go forward? And yes, the fatigue is there, so it's like, okay, do I lay down and take a nap, or do I have to go for a walk? Or do I walk my stairs? But with the gut heart health that you addressed as well. Where do we find all this information, or how is it through HealthTree? Is it through certain publications, or where do we get the best education so I don't start tripping over myself and doing things wrong and then have to re-educate myself?
Yeah, that's a very good question. I think this whole field of the microbiome, as I alluded to, is an evolving area. I can tell you that since I've been looking at it on my Google feeds, I'm getting more and more of these things. And I would start off with just sort of Googling, microbiome and how to maybe improve the microbiome. But I think in general, like I said, the thing that's out there is that animal fats and processed foods and foods rich in artificial sweeteners and things like that are not as good as healthier, natural raw vegetables and more of a heavy, less red meat intensive type of a diet. But I think the internet probably has a lot of that. Greg, wherever he is, I don't know. But certainly I think HealthTree, that probably would be something to begin to look at, because I could see that these kinds of, dietary modifications are things that I'm sure all of us are asked, and it's relatively simple, and to make some of these changes. Just like I also tell patients, don't necessarily deprive yourself of something that you enjoy. Just don't do it in excess. If you like a glass of wine or a little bit of whatever, it's not going to harm you.
And I would, with our foundation, the best point of access for everything is the web page, the homepage of our website. And in fact, we now have an article. There's a study in HealthTree that's being done by Dr. Hillengass at Roswell Park in Buffalo, where he is now working to try to quantify and understand just exactly what effect exercise has on therapy.
Just to second that, physical activity and adequate hydration are sort of the two most important things that we often advise our patients to make sure to stay on top of, and that often gets ignored in the post-transplant settings. I would certainly encourage you to try to be as active as possible. It's good for your bones, and is good for your immune system.
I was unable to get stem cell because I couldn't produce enough stem cells. Is that an issue with T-cell transplants?
Yeah, so that's a great question. The majority of patients will be able to collect enough stem cells. In general, for stem cell transplant, the risk for not collecting enough stem cells is the amount of prior treatment that you may have had. And that's why we try to collect stem cells early. So 95% of people will be able to collect enough stem cells. The stem cell collection is dependent on the mobilization of stem cells from your bones into your blood. When we collect T cells for CAR T, it's dependent on a different cell, a lymphocyte. And typically those lymphocytes are in prevalent numbers in your blood. They don't have to be mobilized from the bone. Now we know that some chemotherapies, if you have exposure, and some of these chemotherapies are given in the treatment of myeloma, usually later on, they can deplete lymphocytes. So we try to avoid chemotherapy that depletes lymphocytes. The chemotherapy that affects your stem cell collection and production, the Revlimid, that doesn't necessarily affect the lymphocyte numbers later on in the ability for CAR T cells. But the number of therapies that you have, and any therapies that kill lymphocytes, can affect your ability to collect CAR T cells.
We also are getting, if anybody has questions, just give me a signal. We're also getting questions from our online audience, and thought we'd ask a couple of those. There are a few questions. Well, let's stick with the theme that we've just been on, and this is a question we get a lot. Is there any scientific evidence about periodic fasting as inhibiting cancer progression? This is a remarkably common question.
The short answer is yes. Short answer is there is data for periodic fasting in the context of, in terms of its effect on cancer progression in certain animal models. Whether or not we can recapitulate that in the context of real human clinical cancer is something that we haven't really established. There are many unanswered questions about that as to exactly how to translate that to the the setting. We certainly are, I think it's what's an open question, however, is can we actually get to a stage where, because there's, our bodies, in general, there's a competition going on between, for the same fuel, we all live with we have, whatever fuel we eat is being competed for by all kinds of things, by the cancer cells, by our normal cells in the body, and by the bacteria that live with us. And so that competition is probably altered in different direction based on what we eat and how we deal with it. Understanding the underpinnings of that competition is something that's a lot of people are studying at the present time, but might actually impact how we deal with some of these questions.
We've had a couple of questions about allogeneic transplants, and of course now I just lost it. Where did it go? Is there still a role for allogeneic transplants, or is the risk-reward equation not favorable? And then additionally, do allogeneic transplants have a role in the rescue of patients after several relapses? What is the likelihood of long-term exposure after an allo? And perhaps for the new ones, just briefly explain what an allo is.
Certainly. So again, this is a question that often comes up. So, we talked a lot about autologous stem cell transplant, which is taking your own stem cells and giving it back to you. People might argue that's really not a transplantation because we are really not taking anything from anyone else. It's your own cells. And many of us would prefer to use it as high-dose chemotherapy and stem cell rescue. You're essentially rescuing your bone marrow by using your own stem cells. Now, allogeneic stem cell transplant, which is again often used in the context of leukemia and other blood cancers, is dependent on getting the stem cells from someone else who is what we call a match or a close to a perfect match, where you're looking at some of those proteins called HLA on the cells. Now, the advantage of using the allogeneic stem cell transplant is twofold. One, you are taking the bone marrow or the stem cells from somebody who is free of cancer, so there is no fear of contamination of the stem cells with the tumor cells. And the second is, you can argue that allogeneic transplant is probably the earliest form of immunotherapy, because those cells that are coming in from the donor is looking at your cancer cell and saying, hey, this is something foreign, and I can go after it and get rid of it. Now, the problem with allogeneic stem cell transplant is it can be associated with significant side effects. Not only does it see the tumor cells as foreign, it also sees your own body as foreign and essentially attacks the body, and you develop what we call a graft versus host disease. And that can have, again, if it's severe or if it lasts for a very long period of time, can have significant impact on quality of life, a lot of complications, increased risk of infection, and a lot of morbidity associated with that. So because of those kind of side effects, we don't routinely use allogeneic stem cell transplant in the setting of multiple myeloma. And so far, there have not been any randomized clinical trials showing that the allogeneic stem cell transplant is better than autologous stem cell transplant if you take everybody with multiple myeloma. Now, having said that, I think there is still a role for allogeneic stem cell transplant in highly selected patient population. So what are those, or who are those people? Now, we talked about all these exciting therapies that can keep the myeloma under control for 10, 15, maybe even 20 years with some of these newer treatments, but many of those treatments, the myeloma does break through. So if you have somebody who is 45 with the diagnosis of myeloma, especially if they have what we think are high-risk multiple myeloma, none of the therapies we have now is likely to keep that under control for more than 10 or 15, at the best maybe 15 years, or probably less than that. So for those individuals, I think it's reasonable to think about allogeneic stem cell transplant. And even in those individuals, I think most of us would prefer to use the current standard treatments we talked about in the beginning, including an autologous stem cell transplant. And if the myeloma comes back fairly soon or sooner than what we anticipate, those are the patients where we would like to consider doing an allogeneic stem cell transplant. Now, one of the questions that Greg pointed out, should we be thinking about it as a rescue after 5 or 6 different therapies? I would say no, because if you use it so late in the course of the disease, it's unlikely to be really successful. So if you're really thinking about using allogeneic stem cell transplant, it's really for the younger patients in whom the myeloma is behaving very aggressively or expected to behave aggressively based on the genetic changes that we see at time of diagnosis.
Another online question. For patients who express the BRAF mutation, what is the experience with BRAF and MEK inhibitors? What tends to be the duration of response?
I'll take a stab at that. So I think the best data we have from a prospective trial is something that was presented at the ASH meeting last year called the Burma trial. So basically they looked at a combination of two inhibitors, small molecule inhibitors, which basically target what we think are the downstream events that happens from that, from getting that BRAF mutation. And they were able to show that a significant number of these patients actually had a response to therapy. What we don't know is how long those responses will last. There have been previous experiences as well, where they have looked at drugs like similar to what has been used in melanoma, where these mutations fairly common, and have shown that the myeloma protein goes down, the tumors shrink in size. Again, most of these responses tend to be fairly short-lasting with just using one drug. And I'll talk about it later this afternoon about maybe some of the clinical trials we are doing, where we are starting to combine these targeted therapy with the standard therapy we use for myeloma to see if we can kind of increase the duration of response from these treatments.
This is a cute question, so I'm going to ask it just as it's written. I just had a transplant, consolidation, and now in maintenance. Doing great. I am 80 years old now, perhaps the oldest patient to have a transplant. In the future, would my age disqualify me from this treatment?
So I think that at one time Medicare, in certain parts of the country, including ours, had an age cutoff of 78 years for autologous transplant. We did not do any transplants beyond that age for that reason. But now there is no age cutoff per Medicare rules. Having said that, we haven't ventured to do transplants in too many people beyond that age. We've considered it and talked to a few patients selectively about it. And I think that it is something that could be considered if you're physiologically young. But I think at some point we probably will say that it's probably not worth the unknown of the toxicity. Because yes, a lot of people do well, but unfortunately there will be some that will not. On that note, probably CAR T cells will probably be applicable to an older demographic. And we certainly have done a lot of CAR T cells in different diseases, even in people in their 80s.
That question was from Stephen. Is he perhaps the oldest patient to have a transplant? What's your experience in terms of an 80-year-old patient? Have you transplanted older patients?
I'm not, but I think there have been patients in their 80s who've been transplanted.
Yeah, no, so we did transplant, I think, 80-plus-year-olds at Arkansas way back when. Yeah, I do remember patients who were over 80 who transplanted before. They did well.
So getting into the fasting that was mentioned earlier, from my limited understanding, autophagy, if I'm saying that correctly, is the sweet spot's 48 to 72 hours. That's been proven to kill precancerous cells. Is that something in fasting that could kill cancerous cells? And if you were to go into fasting, being in remission, would 48 or 72 hours be safe to fast while still taking Revlimid?
Yeah, so I think we have to be a little bit careful in terms of interpreting the existing data and translating it to a clinical recommendation. Because as I said to you earlier, there is data for these kind of approaches in mice in particular to try to protect mice from cancer. But the kind of stuff you can do in mice is different than what would be ethical or would even make sense in the context of people, and to really translate that one-to-one is not that straightforward. So that's the shortest answer I can give you for that. I think translating, let's say for the sake of argument, a 72-hour fast, in a mouse to a 72-hour fast in a human is not the same. And so there's no question that that weight loss, for example, is beneficial towards many health outcomes, including against helping with cancer, but also cardiovascular mortality and other aspects of health. Diet-based weight loss strategies therefore help all those kinds of things. But there's no one single approach that applies to every patient in that regard.
Leah asks, if you relapse on a bispecific therapy, is there any data about responding deeply to another immunotherapy like CAR-T or ADC or another bispecific?
I mean, I would, depends on the target. For bispecific therapies, I spoke about BCMA as a target, but we heard about that there's 3 or 4 others. So I don't know if anybody has gone through multiple bispecifics, to be honest, and enrolled in serial studies like that, but I would hypothesize that somebody who goes through one bispecific has a response to the bispecific, doesn't necessarily predict a response to another bispecific, but could be considered for bispecific therapy. For CAR T cell failures, there's probably a number of reasons that CAR T cells fail in patients. Maybe it's they don't persist, maybe the immune system has, or the cancer has figured out how to evade the CAR T cell. That BCMA antigen still remains on myeloma cells after CAR T cell therapy, so coming in with another therapy might work despite failures of one of the immunotherapies or another. I'd be interested to see what others think though too.
So there are some trials which are actually including or allowing for patients on prior BCMA-specific therapies to go on to another BCMA therapy, whether it's CAR T cells are allowing prior bispecifics or whatever. And the mechanisms for resistance in immunotherapy in general are very, very varied. The mechanism for a targeted resistance could be what we call antigen escape variants, where, for example, in this case, all of the BCMA-expressing tumor cells are killed by the bispecific, and the cells that grow out may be BCMA negative. Now, that's certainly been shown as a mechanism, but it's not the sole mechanism that's been seen with a lot of these patients relapsing. But also the other thing is that a lot of these BCMA-specific antibodies are not necessarily targeting the same molecule on the BCMA, and so there is evidence to suggest, or at least there's a rationale to try other ones. But immunoresistance in general is not a uniform problem or pathway, and so other immunotherapeutic targets should be tried and may have a high likelihood of working.
Yeah, I would just add that, as he said, there are different mechanisms of resistance. If there's loss of target, I think then obviously targeting the same, trying to target the same antigen may not be very successful. But if the target is still present and you have a different mechanism of action, it could very well work. I've had patients who actually progressed through CAR T cells to BCMA and have responded to the antibody drug conjugate to BCMA. Anecdotally, I will also say that, yes, I've had patients who've gone from one bispecific to the other. I've had 3 patients, and unfortunately, these patients didn't respond to their first bispecific, and they did not respond to their second bispecific either. So suggesting that maybe T cell exhaustion, as was being discussed, is a probable mechanism in such patients. But again, these are anecdotal 2-3 patient experiences, and you can't draw major conclusions from these.
Yeah, but the only thing just following up on what Ivan just said, there is that one subset of patients that relapse following BCMA that have this loss of the protein itself because of an actual genetic loss. It's perhaps good to know that it actually has happened or not before you go to another BCMA target. But that appears to be a minority of patients. But it's relatively simple to find out.
Jenny just texted me and wanted me to share with you that we are planning a HealthTree study within HealthTree with Dr. Urvi Shah from Memorial Sloan Kettering, which is going to look at smoldering myeloma patients and test their microbiome samples to see if there's a connection between those. So that's one of the studies that you might be interested in as you as we talk about that. Another question we have online. What is the data and/or clinical experience with a second autologous transplant if the first auto led to a 3-year remission?
As with all treatments, we can certainly reuse autologous transplant too in patients with myeloma. So there have been smaller kind of randomized trials that have been done showing that there's benefit to using what we often refer to as a salvage or a second autologous stem cell transplant in patients who have relapsed after the prior transplant. The one thing that always, or the most important factor is how long did the first response last. So in general, what we have seen is if patients who get one autologous stem cell transplant and don't get a maintenance therapy, and if their myeloma comes back within 18 months of that first transplant, the results with trying to do the same thing again is not very good. And now with maintenance therapies, now we have average time the transplant response lasts is close to 4 years to 5 years. So if someone has a transplant and gone lenalidomide maintenance or Revlimid maintenance and the response lasts less than 2 to 2.5 years, there's probably not much role in doing a second autologous stem cell transplant for treatment of myeloma. However, one thing to keep in mind is there might be other reasons why we may want to do a second autologous stem cell transplant in someone who already has collected cells and it's sitting in the freezer. What we are increasingly seeing is as patients go through multiple different treatments, their bone marrow reserve gradually diminishes to the point their blood counts are really low and they cannot get on a clinical trial, they cannot go on any standard treatment because the bone marrow just cannot tolerate these treatments. In those situations, we have used a second autologous stem cell transplant to reset their bone marrow so that now their blood counts are better and they can go on to a clinical trial or get other treatments that they otherwise would not have been able to.
Any other questions? And then there's one. Okay, let me go back there. I didn't trip, so that was good.
This one's a little, might be slightly off topic or a little remedial, but I'm trying to understand, where does the myeloma cells come from? Do they self-replicate or do they come further up the chain? Ultimately, if theoretically you were able to get rid of every myeloma cell in the body, would you be cured, or would there be latent genetic defects in stem cells or B cells that would cause it to come back?
So that's a very interesting question, and actually, I think we have many answers, not necessarily all answers. We do think that myeloma originates from what we call a post-germinal center B cell. What that means in simple terms is that if you track the evolution of B cell through immune response, it has gone through a process called germinal center reaction, which is what nature uses to make your antibodies get better. And so a cell that's probably beyond that time point is what is the source of the myeloma. However, there are steps between that cell and a real mature plasma cell, because we identify myeloma currently as a disease of plasma cells. And so there are steps between those things that could well be involved in that. Having said that, that's what the source of the myeloma is. It is certainly possible, and there's precedence for similar biology in other blood cancers, that there are abnormalities that occur even earlier in the stem cell compartment. And this is in context of the comment about the risk for other malignancies and things like that as well. What we are beginning to appreciate is that we probably carry mutations as healthy individuals in some of these blood cells. And while we don't understand the natural history of all those mutations, some of those mutations could actually impact the risk of developing that subsequent gammopathy, if you will. I know it sounds complex, But it is probably something that happens in all of us, in all our tissues, not just for myeloma or gammopathy, but we all carry mutant cells. I likely have mutant cells in my body right now that I'm not aware of.
The other, I think, evolving and interesting aspect of this is the microenvironment. In other words, there is a lot of evidence to suggest that the plasma cell itself can't just set up shop randomly in a bone marrow, that there have to be a series of genetic and structural alterations of the bone structure itself in a specific niche that basically allows this plasma cell to travel from point A to point B to establish itself to then spread. And so it's really a very interesting interplay. And in fact, in terms of establishing targeted therapies, not only are we trying to kill the plasma cell, potentially the post-terminal B cell that Madhav just alluded to, but also how to regulate and to render the bone and the bone marrow microenvironment less hospitable to allowing for these plasma cells to survive.
Again, I think you have to also think about it in the context of the time it's taken to get to myeloma, right? We know that, at least based on the studies, it takes probably about 15 to 20 years from the time that early mugger starts to the point the myeloma actually develops. And probably there's some time before that, some of those changes leading up to the muggers itself happened. And we know there are some changes that we see in myeloma that we see even in muggers, and there are some changes we don't. So obviously it's a multiple step process. And the big question I think for all of us is that, do we really need to get rid of every single abnormal cell, or can we just turn the clock back sufficiently long enough that it again takes another 15, 20 years to get to that point, which, again, for a lot of people who are older, that may be the cure.
I think the other aspect of it is we tend to think of cancers as one cell has all the same genetic changes. That's a clone, or they make all copies of themselves. In myeloma, in other blood cancers, it's not just one clone. If we could get at what the founding clone or that founding cell that gives birth to all these cancer cells, potentially that could lead to cures. But in myeloma, what we observe through treatments is that different clones can arise. You can get new mutations in the myeloma. So it's one therapy works at this time, it may not work in the future because the myeloma, the cancer has changed. It's undergone clonal evolution.